12, DNA and genes. So we're going to talk about that. First, let's look at the DNA molecule. We call it the molecule of heredity. And in 1952, Hershey and Chase demonstrated that it was DNA that was the genetic material that dictated the traits, etc., versus proteins, because that's what they had thought it was prior to that it must have been a protein that controlled that. And they demonstrated that it was DNA, not a protein. It is a nucleotide, and nucleotides, remember, are monomers of nucleic acids, and we learned that back when we talked about macromolecules earlier in the year. It's composed of 3 parts, and if you look at the structure on the right-hand side, you can see the different 3 parts, and we'll talk about each one of them. The first one is it's made up of a simple sugar. It's called deoxyribose. That's where we get the D. And the sugar is down at the bottom, and you can see that it is a 3D ring shape. It's made up of a phosphate group, which is right to the left of it. It has phosphorus in it, and then it has the oxygen bond, the bonds of oxygen around it. Lastly, it's made of a nitrogenous base, and you can see that up in the top. The base is there, and it has the nitrogen around it. Those bases are adenine, guanine, cytosine, or thymine. So when we're talking about replicating DNA later, and we're talking about A bonds with T, joins with T, and C joins with G, these are the 4 things we're talking about. And these all are the nitrogenous base of this molecule. So when we talk about adenine, we're talking about the part that is right up there of that base is adenine. And then we can have some with cytosine, some with thymine, some with guanine. Nucleotides form chains when the phosphate groups of one nucleotide bonds to deoxyribose sugar of another. So these nucleotides are going to form very, very long chains, and the chain forms by the bonding of the sugar and the phosphate, and that is going to make the outside layers of our ladder of DNA. So let's take a look at our DNA here. Here are, on the left-hand side, is our DNA molecule. Deoxyribonucleic acid is what it's called. And if you look at the outer 2 rungs, if you look at this as if it was a kind of odd-shaped ladder, the outer edges are the outsides of the ladders. And that is the phosphate sugar bonds that we just got done talking about. That is what makes out the outside structure of our DNA. It's the sugar and the phosphate, and we call that the backbone of the DNA. In the middle are the nucleotide base pairs. And if you notice, adenine, which is A, bonds with T. They always bond together. They spell the word at, so it makes a word. The other 2 are G and C, and so since they're not A or T, they're going to bond together. Those bond in the middle and make up what we call the rungs of the DNA molecule. Those nucleotides are held together with hydrogen bonds. So those bases are held together with hydrogen bonds. Okay? Now, the whole nucleic acid, so look down here at the bottom left, the whole nucleic acid is the phosphorus, the sugar, and either the guanine, thymine, adenine, or cytosine. That whole molecule, that whole section there is the nucleic acid. And so DNA is just made up of pairing of these nucleic acids going straight up. And that's what makes up the basics of the DNA, of the structure of it. So, in 1953, Watson and Crick came up with the structure of what DNA was. And they proposed that DNA was formed of two strands of nucleotides that formed together like the picture that we just looked at. Said that the nitrogenous bases of these strands would pair with each other and form the ladder-like structure that's in the middle of the ladder, those rungs, and it would have that hydrogen bond in the middle. Now, Watson and Crick were able to do this research because there was a female scientist, Franklin, Rosalind Franklin, who actually took an X-ray of the DNA molecule. And that X-ray was what allowed these guys to go ahead and do their research and come up with what they consider to be the DNA molecule, or the structure of the DNA molecule. This ladder is then twisted into a shape called the double helix. So it would be kind of like putting your arms up together and then twist them around each other. Okay? Now remember that this molecule is very, very long, and you have one in every nucleus of your cell, and you have trillions of cells in your body. So we have trillions of these in our body, but they're very, very, very large, and so they have to be twisted and compacted together to fit into the nucleuses of our cells. So here's a picture of Watson's and Crick's model of DNA, and you can see how it is twisted together. But if you look at a magnified portion at the bottom there, you can see where the backbone is, and then you can see where the rungs are put together in the middle and held together by hydrogen bonds. So this is basically the structure of the DNA molecule portion of our lecture. And with that said, I would like you to go to GradPoint and look at the Chapter 12 Miscellaneous module, DNA RNA protein synthesis. Then in there's a Khan Academy. I would like you to take a look at that and look at the structure of the DNA video that's on there. Just look at the structure of the DNA video, and then please check in with your teacher so that she can mark off that you have done your notes and that you know what the big concepts are.